Okay, let's get started. Welcome to the National Alopecia Areata Foundation's webinar, Genetics and Alopecia Areata, What We Know. Joining us today is Dr. Leslie Costello-Socio from the National Institute of Arthritis and Musculoskeletal and Skin Diseases in Bethesda, Maryland. I'm Lisa Anderson, Senior Director of Research for NAF. Before we start the webinar, I'd like to cover a few housekeeping details. We've disabled chat for this webinar session. Please post any questions you have for Dr. Costello-Socio in the Q&A section. Please keep your questions as general as possible for the benefit of all of our audience members. This webinar is being recorded, and all registrants will receive a link to the recording via email sometime tomorrow. And finally, please share your feedback with us. After the webinar, a link to a short survey will pop up in your browser window. Please complete the survey there. This webinar is part of NAF's You Are Not Alone Education and Empowerment webinar series. NAF gratefully acknowledges the support provided for this webinar by our partners Pfizer, Eli Lilly and Company, and Sun Pharma. Thanks for joining us today during Alopecia Areata Awareness Month. Before we get started, I'd like to tell you a bit about NAF and our mission. The National Alopecia Areata Foundation is the leading advocacy organization for alopecia areata. NAF's mission is to drive research to find a cure and accessible treatments for alopecia areata, support those impacted, and educate the public about the disease. NAF's vision is an empowered community with a choice to embrace or live free of alopecia areata. To learn more about NAF's support resources and research and advocacy activities, or to join us as an advocate or supporter, or join the Walk for Alopecia taking place this Saturday, September 28th across the country, please visit our website at naf.org. I'll share some more information about the walk at the end of this webinar. And now on to today's webinar, Genetics and Alopecia Areata, What We Know. We're very pleased to have Dr. Leslie Costello-Socio with us here today. Dr. Costello-Socio is a faculty member at the National Institute of Arthritis and Musculoskeletal and Skin Diseases at the National Institutes of Health in Bethesda, Maryland. She serves in multiple roles there as an associate research physician, as the co-director of the Translational Dermatology Unit, and as a staff clinician on the National Institutes of Health Dermatology Consultation Service. Dr. Costello-Socio's research covers the natural history of pediatric alopecia, including epidemiology, co-occurring conditions, and responses to therapy. She specializes in the use of imaging tools and develops guidelines for pediatric skin diseases. She also studies the microbiome of autoimmune disorders. We're very honored to have her as a member of NAF's Research Advisory Council and to have her with us here today. So Dr. Costello-Socio, thank you so much for being here, and I'm going to stop sharing and turn it over to you.
Thank you, Dr. Anderson, for the invitation. It's really a pleasure to be here, and really a pleasure to see so many people interested in the genetics of alopecia areata. I'm going to share my screen. Okay. So one of the most common questions that I get from patients when they're diagnosed with alopecia, or if a family member is diagnosed with alopecia areata, is this a genetic condition? The answer is tricky. So if we look at the most simple answer, we say yes, it's genetic. 1 in 5 people with alopecia areata, and when I say alopecia areata, I mean alopecia areata spectrum, which includes universalis and totalis, has a family member with AA. And we know that certain genes increase the risk, but if you have these genes, you don't necessarily get alopecia areata. The more complete answer, and what I'm going to kind of discuss through this talk, are that there are other characteristics that influence autoimmune disease. In addition to DNA changes, which are super complicated, environmental factors like diet and smoking and where you were born or where you grew up, infections, and many unknown things have an influence on our genetics. So why have we suspected that alopecia areata has a genetic component? So for most diseases, the first thing that's done is something called twin studies. So looking at identical twins and fraternal twins and asking, is there concordance between one twin and the other in terms of disease? In the top is a picture of two monozygotic twins, so identical twins. They're sometimes, in this case, referred to as mirror twins because one is right-handed and one is left-handed. And if you look at their alopecia areata spot, you can see it's almost on the opposite side of their head. And below is a study that NAF did in 2010, looking at monozygotic twins. And you can see that the concordance rate was 42%. So what does that tell us? It tells us that there's likely a genetic component because that's more than just chance and more than what we see in the general population. But as we'll talk at the very end, it tells us that it's not 100%. So there are other factors that influence whether you get alopecia areata. In contrast, if you look at dizygotic twins, which are fraternal twins, you see the concordance is only 9%, which is more similar to family members or first-degree family members. So parents, siblings of a person who has alopecia areata. Second thing we know are there high rates in some families of alopecia and other autoimmune diseases. So first-degree family members have an estimated risk of about 7 to 8% of developing alopecia areata compared to the general population, where the prevalence is about 2%. And there's this concept of like familial autoimmune disease aggregation, where there are specific loci, so these are susceptibility genes, and we'll talk more about them, that put you at risk for a number of autoimmune diseases. And the third thing that makes us know that this is genetic is that alopecia areata is seen in very high percentages in some primary immunodeficiency syndromes. So these are syndromes where you have an immune defect and you have effects from that immune change on multiple parts of your body. So this is something called autoimmune polyendocrinopathy syndrome, where about 30 to 40% of patients develop alopecia areata, usually within the first 5 to 10 years of life. But they also develop candidal infections, they develop Addison's disease, they develop autoimmune hepatitis. There's a number of these inborn errors of immunity, so that's the category of these primary immunodeficiencies that are seen with high rates of alopecia areata. And you can see from this pie chart that there are defects in many different areas of the immune system. And we see in these patients anywhere from 10 to 40% chance of alopecia areata, in addition to other autoimmune diseases. So does this mean that all alopecia areata is caused by a single gene? So the quick answer, and you probably have guessed, is that I've shown you, multiple genes that are different. Some of them are in the same pathway, but many of them are not. And these, about only 10% of cases have alopecia areata. Whereas in 90% of patients with alopecia areata, they don't have any changes in these genes. So to really understand genetics, we have to understand a couple of things. So monogenic diseases, so these are like those inborn errors of immunity. You have a single change in your gene, and that leads to a global immune defect. And that kind of change causes lots of systemic problems. And one of those can be alopecia areata. For the majority of patients with autoimmune disease, they have polygenic, meaning there's multiple genes that cause their trait or their phenotype. And for some, it's complex because it's not just the gene, it can be other things that influence genes. So for those of you that remember your high school biology class, you might remember Gregor Mendel. He was a cloistered a clergy person who did experiments with flowers and plants to understand genetics. And this is the perfect example of monogenetic disease. So you have, a parent that has one gene and the other parent has a different gene. And when they combine, you get a different color. But then if you combine two kind of parents that then have this A, then you can get a range of colors. So here, the trait is really determined by one gene. And that's the case with these inborn errors of immunity. So for the autoimmune polyendocrinopathy syndrome, there's a change in this AIR gene, and that leads to a failure of T cell tolerance. And so what I mean by that is that the T cells, which usually don't recognize self, don't create that ability. And so as a result of that, patients who have this gene mutation develop a lot of autoimmune complications. They also develop complications from infections because they don't know how to fight those off. So is alopecia areata really different in these inborn errors of immunity? Often it occurs in childhood. So, many of you had your first patch of alopecia areata when you were a kid. Some people don't have it happen until they're young adults or middle-aged adults. But mostly you can't tell any difference except that you might have other symptoms. You may have recurrent infections. You may have more than two autoimmune disease. And there's been kind of an explosion of kind of work on these inborn errors of immunity with alopecia areata, which is really important because it's teaching us a lot about why patients that don't have monogenic causes of disease have their diseases. So this is an example of something that my group worked on. So we looked at age of patients and when they developed alopecia areata, vitiligo, and hypothyroidism, and their first manifestation of that autoimmune polyendocrinopathy. And you can see in blue that by the age of 12, more than 80% of patients already had alopecia areata, which is a little bit different than what we would see in the general population. And then you can also see these patients then went on to also develop vitiligo and hypothyroidism. So this monogenic disease is important because it really teaches us, why does this disease happen? We learned a lot about like T cell tolerance. So, knowing that this is a failure of, seeing yourself. And it also taught us a lot about how infections and other stressors may impact alopecia areata. So having, multiple infections, having other autoimmune diseases may influence when you develop your alopecia areata or the severity of the alopecia areata. But most autoimmune disease, including alopecia areata, is polygenic. So, this is pumpkin pie season. We have, 10% caused by monogenic. And so that's more simple to explain, right? So you have a change in your immune system that's very discrete and you get alopecia areata. But in the rest of slices of the pie, multiple genes are contributing to this trait. And this is more similar to, complex traits. So these are twins that were in the news a few years ago because they look so different. They have different texture hair, different color skin, different color eyes. And it tells us a little bit about how hair color, eye color, skin color is not really like the Mendel kind of paradigm where one gene is responsible, but here multiple genes, lead to whether you have, blue eyes or brown eyes. And so this is an example of like non-Mendelian genetics. So things get a little bit more complicated. So, your parents input multiple genes each. So let's say, the biological mother on the top and the biological father on the side. And depending on which gene they input, you have, different color skin or different color hair. So here the trait's determined by more than one gene. And some of these genes might not even be coding genes. So I'm going to talk a little bit about what that means and why that's important for alopecia areata, especially when we're kind of coming to this age of wanting to test for the gene change. So autoimmune disease is complex inheritance. So when we look at our DNA, we have exons. And so these exons are what we call coding regions. So the letters that make up the DNA make a protein. But between these exons, we have introns, and these are non-coding regions. So these kind of can influence how the gene works. These might add something on to certain proteins. These are different than just, simply you have a bunch of letters, that letter equals one protein. And these become pretty important when we talk about alopecia areata. So when I say things are complex, we start to understand that there's additive effects of multiple susceptibility genes, and I'll show you how we kind of figure out what those are. We have these things called single nucleotide polymorphisms. So these are single changes in a gene that may not impact anything about the structure of the protein, or they might just influence a tiny part of that protein. And if you have a single one, nothing happens. But if you have multiple, they can add up. We know that these SNPs, or single nucleotide polymorphisms, can lead to variation in how a gene acts. So we could have high levels of the gene or low levels of the gene based on these small changes, so often in these non-coding areas. And then, of course, something that I think many families and patients are interested in is, how does our environment act on our genes? And, are the things that we're doing or living through affecting, whether we develop an autoimmune disease or not? So the environment may change the expression of genes. So when I think about single nucleotide polymorphisms, I think about those highlights magazines. So I might be dating myself, but when I was younger and I went to the dentist or the pediatrician, we would sit in the waiting room. There were no TVs in the waiting room, but there were always highlight magazines. And for many kids, one of the most fun thing was to spot the difference. And so this is, to me, the exact replica of what a single nucleotide polymorphism is. So let's look. So you have a tree, right? So this tree kind of has a flat kind of top, and this tree has kind of, extendable leaves. Now, they're both trees, and we would look at these and call this tree. But if it rains, this tree might work better at providing coverage than this tree. If we look at the little hippo, she has a green helmet, and this hippo has kind of a red helmet. Now, this little change might have zero impact on our picture or her ability to bike. And then if we look at our little lion here, we see, he's on a unicycle. And on the unicycle, he has one big wheel and one little wheel. And while this is still a unicycle, if we were thinking about the race that these animals are in, this one with the bigger wheel might have an advantage over this one with the small. So single nucleotide polymorphisms are these little changes that, by themselves really don't have a huge impact. But let's say, you have a difference in a wheel, and then maybe there's a rock in front of, this wheel that could really impact the outcome of the race, or for us, the outcome of the gene expression. So, how did we figure out, about these SNPs and about, loci in big populations of patients that don't have inborn errors of immunity? So Angela Cristiano, which many of you know, is a professor of genetics at Columbia, did genome-wide association studies for patients with living with alopecia areata and patients who did not have alopecia areata. And she compared the DNA of those patients to each other to see, what's the frequency of different SNPs. And, do these appear in all patients with alopecia areata? Do these appear in a high, frequency? Uh, and so then she used this data to carry out like analysis to say how likely is this variant associated with the trait. And so this was important because it was really the first time that we saw specific genes that were associated with alopecia areata, and most of these were these single nucleotide polymorphisms. And many of them weren't even in coding regions, but they had an impact on whether someone had alopecia areata, and perhaps even if someone had patch stage alopecia areata or alopecia universalis. And from this, she identified a number of immune system related genes, which probably hard for you to remember, but many of these are the same genes that we saw in the patients who had inborn errors of immunity. So, genes that are involved in T cell activation, genes that are involved in immune tolerance. We also found hair-related genes, so genes that are related to hair pigmentation might explain why when we regrow alopecia areata, it comes back white. Some genes that are, responsible for hair shaft integrity, maybe explaining why we get those exclamation point hairs. And then there's a number of other genes that were involved in developmental processes and antioxidant defense. And so, across the world, so this was the study that was done in the US, this study was done in Europe, and this last study, Yang, was done in Taiwan. And what's like beautiful about this is once you do these genome-wide association studies, you can compare the changes that you find with other diseases. And I always tell the students and residents that this is one of my favorite diagrams of, for alopecia areata, because when this was first published in 2013, it really gave us a sense of the connectivity between autoimmune diseases and AA. So you can see, here's AA and some of the genes that were different in the genome-wide association studies. And then you can see other diseases like type 1 diabetes, systemic sclerosis, rheumatoid arthritis, celiac disease, IBD, and they're all kind of like clustering around and sharing a lot of these risk loci. And this is, I'm not going to talk about JAK inhibitors, that's another talk, but this is what really led to the idea that maybe if we inhibit the JAK-STAT pathway, which is responsible for some of these differences in the immune system, we might reverse hair loss and have new hair growth. So, can we easily test genetics of alopecia areata in clinic? So I have a natural history study, and as part of that, I do, whole genome sequencing of anyone who comes to see me for that study. And I always tell patients at the outset that the majority of patients that I see won't get an answer from that. They're going to have what's called a negative genome, meaning that the biologists who were looking at the sequences looked really carefully at it and didn't see any differences, and so they say we didn't find any reason for the alopecia areata. The reason to do this is to find those 10% monogenic diseases. But this is not like perfect testing either. So, both whole exome and whole genome sequencing, can identify those monogenic diseases. Whole genome can identify those polymorphisms, but sometimes we're not sure if they're meaningful or not. And so, when I was thinking about like, how do I explain that? So, this is the Hitchhiker's, Guide to the Galaxy. So if we're doing like whole genome sequencing, we're going to skim each of these books and look for the words and make sure that the words, that the letters spell words. Now, when we're going through quickly, we could make an error, right? So the word there, THERE and THEIR might, they both spell there. And so if we're going through quickly, we might not see the context and we might say, oh, everything looks great. And if we're doing whole genome sequencing, where we're looking at things that are not just coding, we might be looking at, does the font impact the reader's experience? Does the thickness of the paper make it more likely for someone to get a paper cut? But we're doing this quickly, and so we might not notice that that's the case. So the answer is no. Right now, we can't easily test genetics of AA in clinic. And so, I think when I'm, thinking about the science of genetics of alopecia areata, I think, wow, there's been some really meaningful variants that have been identified. We've learned so much about the immune system's impact on, why some people get alopecia areata and some don't. But we need better and more meaningful clinical tests or panels. And then we need to do the second things, which is look at the level of the hair follicle, because maybe it's not just all our genes that are impacting whether we have AA, but it's just the genes that are upregulated or downregulated around the hair follicle. And if we look at these before and after treatment, we might really learn a lot about what's influencing those genes. And then we need to, kind of figure out if there are environmental factors that are interacting with how those genes function, both, in the global level, so the whole individual, but also, down to the cell level, like the T cells that are surrounding and swarming, the hair follicle. So, can we do this? And we're starting to. So there's something called single cell RNA sequencing, which allows us to take tissue like skin and skin that has hair follicles and look closely at all the components that make up that skin. So this is examples of someone who doesn't have alopecia areata. So this is a punch biopsy. So this is a skin punch with some patients may get this with alopecia areata. And this is a patient who has, as you can see, less hair follicles. They have alopecia areata. And then we kind of separate this into the cells that make up this tissue. And then once we do that, we start to analyze the sequences and what's happening in those individual cells. So this is a what's called a UMAP plot. So it tells us, like, we label the cells and say, okay, lymphocytes have this marker on them. Fibroblasts or melanocytes that make pigment have this marker. And so we're able to know where those cells are from that whole biopsy. And then we could say, all right, we're only interested in the T cells or the white blood cells, the lymphocytes. And then we can then start to look at, okay, well, there's lots of types of white blood cells. We can, label them and start to figure out, how they're different in patients with alopecia areata and patients who don't have alopecia areata. And so this is an example of doing that. So HV means healthy volunteer. This is a patient living with alopecia before treatment with a JAK inhibitor. And then this is the same patient after treatment with the JAK inhibitor. So, these are a lot of letters and things that kind of tell me a little bit about each cell. But I think what you can see with the colors is that, compared to the healthy volunteer, there's, a whole different population of cells that are very active in patients with alopecia areata before treatment. And, this green one and this kind of mustard yellow one. But after treatment, you start to see kind of normalization of that number of cells. So now we know a little bit more about, like, okay, these specific lymphocytes are really important, but how are they interacting in the tissue? And can we learn about, why there's so many of them? Is it because they're genes that are being upregulated in those cells or downregulated? And so we can do that by looking at specific genes. So we can tag those cells with genes, and these are different, like collagen genes that are part of just typical tissue. And then look at, these are called violin plots, a healthy control, so someone who doesn't have alopecia areata and someone with it, and start to see, oh, this gene is really, upregulated compared to in patients with alopecia areata. And that can tell us kind of the next step, which is where is that happening? And maybe we can figure out why. So there's something called spatial transcriptomics, which is a way to incubate tissue with gene probes that we learned about from the single cell RNA sequencing, and ask, where are genes up or down within the context of the hair? So these cells are not kind of dispersed. This is a punch biopsy. This is the top of the skin. This is down to the fat of the skin. These are hair follicles. And now we can ask, okay, we saw those genes are up and down. Tell us more about where they are. And so this is really pretty, but you can see, we have probes for predefined genes. So this is like hundreds of genes that we are interested in. And you can quickly see that someone with alopecia versus someone who doesn't have alopecia, their probes mapping looks really different. But this is kind of hard to look at because there's so many genes. So now we can say, all right, maybe we just want to look at 2 genes. And so, here we're looking at gene 1 and 2, and we're asking about, where they're located, within the hair follicle. And here you can see that this like mustard yellow, gene is really downregulated in patients with alopecia areata, where, and whereas this second gene in blue looks about, similar to in the control. So maybe this is, the gene that we're most interested in. And then we can, look more closely at patients with that gene and say, is that gene different in those patients? And often what we find is, no, that gene looks perfect. So, what is controlling whether it's, upregulated or downregulated? So the problem with like single cell and spatial transcriptomics right now is that it takes a lot of time and it costs just a lot of money. So to do like one test, it's like $25,000. So obviously we can't be doing that on every patient that comes in. One, because we just don't know what genes we would be looking for, but also, we need a more practical kind of way to kind of approach, whether a gene is upregulated or not. But it tells us a lot about these non-coding genes and the environment, and how the environment might be impacting the genes that are responsible for leading someone to have alopecia areata perhaps at age 5 versus at age 20 or not at all. So I'm going to go back to twin studies. So these are identical twins. And now these twins are well into their 50s. And even though they were perfectly identical when they were infants and toddlers, you can see with time, they have had environmental things, sun in this case, that has changed the quality of the skin in terms of freckling and wrinkling. So we know that in identical twins, they should be, sharing very similar DNA, but somehow over time, environmental factors impact that, and so the outcome might be different. So there's been work in something called epigenetics. So epigenetics are things that occur to the gene or in the environment of the gene that leads to the gene being different. So these are chromosomes. So this are 3-year-old twins, and these are 50-year-old twins. The 3-year-old chromosome 3, and we're looking at something called methylation, which is important for DNA. they look pretty similar. So at 3, these twins are pretty much identical. That chromosome isn't gonna do anything different. They're gonna have a pretty similar experience. But by the time, these twins turn 50, the methylation pattern really starts to differ. So you see, excess red in this part of the chromosome for this twin versus, more in the center for this. There's tons of methylation in the middle here and not much there. So, there are environmental things that are making people who have identical DNA do different things. So epigenetics, it's changes in DNA that don't involve alterations to the underlying sequence. So this is what I'm talking about for the gene when we're looking at that tissue. We look at that gene and it looks perfect. There's nothing different about it. We do whole exome sequencing, whole genome sequencing, and we get a negative genome that says there are no changes in the gene that would be responsible for this. But that's not the whole story for genes. There are things that happen in utero, in childhood, when we get infections, when with diets that, change little kind of ornaments on our DNA, things that are like methylation or something called a histone tail. And if we have enough of these changes, the gene might not function in the way that we thought it would. It might now not be able to upregulate in the way that we thought it should to protect against self-tolerance. Or noticing that the hair is foreign. And we're starting to see that these are factors that lead to cancers and autoimmune disease, diabetes, mental health disorders. What's fascinating about this field is that maybe in the future, and we're far from that, but not so far, we could manipulate these little changes to make that gene behave like we would expect it to. And we're starting to do that a little bit. So there are histone modifying enzymes for colorectal cancer. So a lot of colorectal cancers have very specific mutations that lead to, cells over multiplying, or they have typical genes, but they have these modifying enzymes that make them kind of overgrow. And so if we can block those enzymes, the cells stop overgrowing and maybe don't metastasize. So, this in the end might be more targeted than, blocking the entire immune system. And it gives me hope, especially when we think about, some of the medicines that we have, which have side effects. And I think things that as a physician, I worry about, and as a family and a patient, I think many patients worry about. Maybe we can do something that is less detrimental to the entire immune system. So is alopecia areata genetic? So it's yes, but a lot of progress has been made. We know that there are risk alleles, and the more you have, the more likely you are to get an autoimmune disease. The more we learn about genetics, I feel like the more we need to know, and it's very complicated. We know stuff is happening at the tissue level, and so when we do these tests of our blood, maybe we're not capturing the differences. And maybe we can use some of these new technologies like spatial transcriptomics and epigenetics to really understand our diseases better. And then this is my email, so I'm happy to always take questions. So you can just email me if you can't. If you don't have time to write this down, we're going to give handouts out. And then I have to say that this talk is just my own opinions on science and not necessarily my employer. And so I'm going to stop sharing and take some questions. I saw some in the chat.
Thank you so much, Dr. Costello-Socio. That was, I mean, it's such a complex topic, and I think you did a really good job of simplifying it as much as possible, but it's probably not really possible. So there's so many, I'm sure there's lots of questions, and let's get through as many as we can. Maybe some of it will, we'll be repeating some of the stuff that you said, but perhaps with a little bit of a different spin on it. So for example, this one, you maybe touched on this, but are the genetics different in adult onset alopecia areata versus early onset? Like, what can you say or summarize?
Yeah. Yeah, that's a great question. So I do think that there are probably differences in the reasons for why someone develops it in childhood versus adulthood. It may be as simple as those who develop it in childhood have more risk loci, and those that develop it later on maybe have other, I don't want to say hits, but environmental things or other things that might influence their genes. And then we know from that kind of smaller 10% group with the inborn errors of immunity that those patients tend to, get alopecia areata at younger ages. I don't want people to worry that if they have alopecia areata as a child, that they definitely have an inborn error of immunity, because the majority of people do not. But it's like a clue if other things are going on.
Okay, thank you. Okay, here's another question. Can these genetic differences among people with alopecia explain why some people respond to treatment better than others? Do we know
Yeah,
that?
so that's a great question. So probably, right? So we know that for the majority of patients, that JAK-STAT pathway is really important for the pathogenesis of alopecia areata. But there are some patients that don't seem to respond to those medicines. And our best guess is that there's a different pathway that is more important for their disease.
Okay, thank you. You touched on this as well, but could you perhaps restate what some of the environmental factors that may affect gene expression are?
Yeah, so I think we're certainly learning a lot about this. So your microbiome, I think, is important, and we're learning a bit more about that. And I know that Dr. Hordinsky's group is kind of looking at fecal microbiome and its impact on autoimmune disease. How, infections probably do play a role, because of course infections do affect your response, how your immune system responds. But I think we're really at early stages, and we don't really know, if you have one infection, does that increase your risk, or you have to have three? So, I think it's things like diet, environment, like, that affects microbiome, and maybe things like global, like where you live, that have impact.
Okay, thank you. Here's another question. Have you done any studies considering the comorbidity of alopecia areata and Down syndrome? And this person writes that 10% of Down syndrome population have alopecia compared to 2 to 3% of the general population. And is there something that we can learn from that dual
Yeah.
diagnosis?
Yeah, there are two groups that are looking at alopecia areata and Down syndrome. So one is a group in dermatology at Dartmouth University, and the principal investigator is named Gillian Rourke, and she has an interest in why patients who have Down syndrome have many autoimmune diseases. And then there is a Colorado cooperative that looks at Down syndrome also, and is interested in alopecia areata and other comorbidities.
Okay, sounds good. Maybe that's a future webinar topic, who knows?
Mhm.
Does the identification of specific genes determine the course of treatment?
Yeah, so not yet. However, for other diseases, we're starting to see that. So, for example, there are something called HLA types. And based on your HLA type, you might be at high risk for like a severe drug reaction to a certain medicine. And so you might not be a candidate for that medicine. And so in those diseases, they check your HLA type before starting medicines. And so I can picture that not too far in the future, we may be doing similar things where, the kind of the idea of that panel where we understand what are the risk alleles and then saying, okay, you have these, so maybe this treatment is better, but we certainly are not there yet.
Okay. Okay, here's another one. I developed alopecia areata at age 70. No one else in my family has ever had alopecia areata. That's from newborns up to this person's age of 70. What could have caused it? So,
Yeah, so that's tricky. So, people like you who develop alopecia areata over the age of 50 are more rare. And my best guess is that you do have risk alleles, but you didn't, like, those SNPs weren't causing any impact. And perhaps having COVID or having some other environmental insult, triggered those susceptibility SNPs to behave differently. And I think, with some viral infections, we certainly see that. With that said, we can't like prevent viruses, mostly, anyone who has, kids or works in a place where there's lots of people, you're going to get lots of viruses. But that's why I think that is the reason that we would want to do vaccines. But it seems like in your case, maybe that vaccine revved up your immune system.
and that's also part of their question is like,
Yeah.
could this have been from COVID vaccination? And then there's another question like, could COVID have contributed from a different person, environmental causes? So it could
Maybe,
having the
yeah.
virus itself or the vaccination, either of those could.
Yeah, so we know that COVID raises level of interferons, which are what is the end product of the JAK-STAT pathway. Most infections do that. So, it would make sense that we might see some differences with having viruses. There have been a number of studies that have tried to look at that and say, were there more AA cases, with the COVID pandemic? And most of those studies they're not super strong, suggesting that it did. But it's probably because it's really hard to do that kind of study.
Yeah. Okay. There's another topic.
Mhm.
Do JAK inhibitors work at the genetic level or on cells or enzymes that are not functioning correctly due to the genetic mutation?
So no, so they're just blocking that immune response. So as far as we know, and we don't know everything about JAK inhibitors because there are what we call off-target effects. We don't think it is correcting genetic changes.
Someone's asking the question, what are some environmental factors that affect DNA methylation?
Oh, that's a good question. Probably a variety of chemicals, plastics, uh, medications can affect methylation, probably radiation, those types of things.
Okay, thank you. Let's see. What is the correlation of hypothyroidism and alopecia both developed in childhood?
Yeah, so the most common second autoimmune disease that patients living with alopecia areata have is hypothyroidism. So there are, shared risk alleles between alopecia areata and autoimmune thyroid disease. Why we see those more commonly in childhood, together, I don't know.
Okay, thank you. Do you observe a connection between eczema and other skin conditions and alopecia? Can those same environmental factors play a role?
Yeah, so I think, the story of eczema is an interesting one. A certain percentage of people living with alopecia have atopic dermatitis or atopic disease. Those patients might have a slightly different pathway for their alopecia areata, and that's why maybe medicines like Dupixent might help their alopecia areata. Um, if we're thinking about environmental factors, certainly we know eczema changes your skin microbiome. And so, perhaps that's, playing a role, but I don't know that we understand skin microbiome in alopecia areata very well.
Okay, thank you. And just a plug for some past NAF webinars. We have had some about the connection between eczema, atopic dermatitis, and alopecia areata. So check back our old webinars for some more details on that. Thanks for answering that, though.
Mhm.
Who is the best candidate with alopecia areata for genetic testing? Are there like characteristics or
Yeah,
conditions? I mean, do you think that is something that will become more common in the future?
yeah. So I think patients that have more severe alopecia areata, patients who have other secondary autoimmune conditions in childhood. So kind of having both happening early, um, frequent infections. For now, I think the thing we can look for is those monogenic diseases. And so I think if we're looking at who's best for genetics, it's those patients who have multiple family members with very similar symptoms and then have other symptoms besides their alopecia areata, and usually have it early in life.
And is that something that is going to be commonly suggested or is it pretty
Yeah,
specialized?
and I think genetic testing isn't covered by most insurances. It's challenging to get it, especially for alopecia areata currently. Um, I think if we are seeing infections, you might be able to get coverage, but I don't think it's going to be a standard when someone comes in for patch stage alopecia areata that you get genetic testing anytime soon.
Okay, thank you. Is stress considered an environmental factor that might have an influence?
Yeah, so one of the changes in the GWAS was this oxidative stress, so stress to cells. I think stress can mean a lot of things. So, emotional stress, stress to the body, from fatigue or illness. So yes, I do think that it is what I would consider one of the environmental things.
And someone wrote somewhere earlier in your presentation, you said where you live may have an impact.
Yeah.
Can you say what you mean by that?
Yeah, so for autoimmune disease, there are starting to be these zip code studies where populations of patients who live in certain areas are being looked at more closely for the diseases that they have. And and this has been done for things like breast cancer and lymphomas kind of in the past. Usually those environmental things have been chemical plants, other things that have gotten into the water, like, chemicals or other into like water system.
Okay. Someone wrote, I saw SS on your slide, the slide with the different autoimmune conditions. Is that Sjogren's syndrome?
Systemic sclerosis.
Systemic sclerosis, okay. Let's see. Is there a difference for those who are African American when it comes to, it says when it comes to alopecia, but perhaps you could, since we're talking about genetics.
Yeah. So, I think we certainly are moving away from defining, people by, race and ethnicity only. because I don't think that's like a perfect surrogate for our genetics. We do know from epidemiology work that I did, and then another group looking at a nurse's health study, that patients who identified as African American or Black had higher rates of alopecia areata. And that was true in the US and the UK. So perhaps there are genetic differences, but I'm always careful because the genetics can be very complicated to understand, like origin for everyone.
Okay, thank you. These are all really good questions and complex, obviously, not easily answered.
Yeah.
So here's another question related to environmental sources and impacts. Someone saying that their daughter was diagnosed at age 7, likely triggered by a viral illness. Is there anything else we can do to reduce the environmental impact?
Yeah. Yeah, so I get asked that a lot. You know, my response is always that we can't really worry about, you know, the infection that happened. We probably, you know, maybe another infection would have done it in 6 months if the first one hadn't. So it's tricky to, I think it's just tricky to prevent yourself from getting infections. And so there's not, you know, other than standard like hand hygiene and, you know, things like that. So I think the short answer is probably not. I always recommend people eat like whole food diet, try to eliminate processed foods. But realistically, I eat processed foods. We all do. And so we can't, you know, we can't also isolate kids by making regimens that are really challenging socially. So there's a balance.
Right. Okay, we're almost at the top of the hour. We'll take a few more questions and then we'll wrap it up.
Okay.
What are your thoughts on a relationship of a parent with rheumatoid arthritis and the child having alopecia areata?
Yeah, I think that that is I think it's a perfect example of like a risk allele that is shared. And the outcome for the parent is rheumatoid arthritis, and the outcome for the child is alopecia areata. We don't quite understand why one is the output is one and one the output is the other, but it is, I think it's related to a risk allele.
Yeah. Okay. I'm just going to look, just scrolling down real quick here. I think I'd like to ask one last question that's sort of buried in many of these, and you touched on it a little bit, but how do you think this research, or this kind of research, how does it affect our treatment landscape looking out into the future?
Yeah, I think that we don't want to just have a single treatment, because we know that even with how wonderful the JAK inhibitors are, there are patients that don't respond. So I think this work does two things. It provides, you know, different avenues for therapy, and then potentially avenues that might be safer for long-term therapy. So I think that those are the, you know, kind of the goals of really understanding, you know, the genetics at a very, very fine level.
Yeah. Well, I think that's a good place for us to wrap it up. Thank you for sharing your expertise with us, and thank you for the research that you're doing in this space, because as you know, this is where the new treatments are going to come from, better understanding of the disease is going to come from. So thank you so much, not just for being here, but for the work that you do. We appreciate you.
Yeah, and thank you everyone for attending. Yeah, I'm very humbled that so many people are, you know, here at this late hour in the East Coast.
Yeah. Yes, thank you so much.
Yeah.
I'm going to share my screen again, and then we will wrap up this webinar. Give me a second. Okay. Thanks again, Dr. Costello-Socio. We really appreciate you. And thank you to those of you in the audience for being here. We hope you share your feedback on this webinar. You will also be helping us plan future presentations. So when we close the webinar, a short survey will pop up in your browser window, and you can share your feedback with us, and we really appreciate that. Now, as I mentioned earlier, the Walk for Alopecia is happening this weekend on Saturday, September 28th. Alopecia Areata Awareness Month is almost over, but it's not too late for you to participate. There's still time for you to make an impact by registering to walk or making a donation. We are at about 80% of on our way to our goal, and you can help us reach it. More than 2,300 people are registered to walk around the country. You can join a walk at one of the 9 sites around the country, or you can walk where you are, where you live. For more information on the walk, you can scan the QR code that's here on the slide, or go to our website, naf.org/walk for more information. We really hope that you will join us. And join us for our next webinar coming up on October 10th, Maintaining Scalp Health for All with or Without Hair and Wigs. This webinar will cover comprehensive scalp care practices to maintain a healthy scalp. Professor Maria Hordinsky, MD, from the Department of Dermatology at the University of Minnesota Medical School will explore a range of valuable tips for maintaining optimal scalp health. Advice for wig wearers and those who do not wear wigs will be covered, with a discussion of the unique scalp care needs for each group. Join us for this presentation that will empower you with the knowledge and tools necessary to promote and maintain the well-being of your scalp. This webinar will take place on Thursday, October 10th at 7 p.m. Eastern, 4 p.m. Pacific time. Registration for this registration for this webinar is now open, and you can scan the QR here to register. QR code. Okay. There's a lot happening at NAF. How can you keep up? Well, you can subscribe to our email list and get regular updates on breaking alopecia areata news and research, our monthly electronic newsletter, as well as notices about upcoming webinars and other programs. If you're not already on our email list, please scan this QR code to enter your email address and receive our emails. And don't forget that NAF offers a number of resources and programs to the alopecia areata community, including support groups, our youth mentor and legislative liaison programs, the doctor finder, clinical trial listings, as well as news and webinar links and how to get involved. To learn more about NAF and the resources we offer, please visit naf.org or email us at support@naf.org. And this concludes today's webinar program. Thank you so much for joining us. Thank you, Dr. Costello-Socio, again for being here. We look forward to having all of you back for our next webinar. Take care, everybody. Have a good night.